Okay, so get this. You sent over a research paper, and the title, well, it's a mouthful. Yeah, it's a high-quality peptide evidence for annotating non-canonical open reading frames as human proteins.
Quite a title.
My head's spinning a little bit, but you know me, I love a challenge. And this deep dive, I have a feeling it's going to be mind-blowing.
Oh, it is a dense topic, but you're right. Very exciting.
Yeah.
We're talking about hidden proteins in our DNA.
Whoa.
Proteins that scientists didn't even know existed until very recently.
It's like finding a secret passage in your house, leading to a whole new wing that you never knew was there.
Right.
These are called non-canonical open reading frames, or NCORFs for short.
That's right. You see, we thought we had the human genome all mapped out like a finished puzzle.
Yeah.
Turns out there were all these tiny pieces just hiding in plain sight.
Oh, wow.
And these NCORFs, these extra puzzle pieces, hold the potential to change everything.
Okay, so walk me through this.
Okay.
What exactly are NCORFs?
So,
Why were they missed for so long?
well, for years, scientists have focused on the parts of our DNA that code for the proteins that we already know about.
Right.
But sprinkled throughout our genome are these stretches of DNA that were considered junk.
Oh.
They didn't seem to do anything.
Right.
But now we're realizing that many of these so-called junk regions actually contain the instructions for building tiny proteins called microproteins.
Wait, so our DNA has been hiding a secret stash of tiny proteins all along.
Exactly. And most of these NCRFs are incredibly short, under 100 codons.
Okay.
To put that into perspective, the average protein is hundreds of codons long.
Wow.
So it's no wonder they were overlooked.
Yeah.
They're like these tiny needles in a giant haystack.
So these microproteins, they're tiny but mighty rights.
They are.
This research that you sent me, it seems to be a big deal in the NCORF world.
It is.
What makes it so special?
You won't believe the sheer scale of this research.
Okay.
They analyzed a mountain of data, billions of data points, from over 95,000 experiments.
Wow.
And they used some incredibly powerful techniques to do it.
Okay, break it down for me.
Okay.
What kind of techniques are we talking about?
There are two main approaches: conventional proteomics and immunopeptidomics.
Okay.
Let's start with the first one.
Go ahead.
Conventional proteomics is like taking apart a complex machine and carefully analyzing each component.
Got it.
Scientists break down proteins using enzymes and then study the resulting fragments to identify them. But this method only found evidence for a tiny fraction of the NCORFs actually being translated into proteins.
So conventional proteomics wasn't quite cutting it for these tiny proteins.
Exactly.
Yeah.
That's where immunopeptidomics comes in.
Okay.
This technique takes a completely different approach.
Okay.
Instead of looking for the proteins themselves, it focuses on the tiny protein fragments presented on the surface of our cells by the HLA system. Think of it as our cellular security system.
Right. The HLA system, part of our immune system.
Yes.
Constantly scanning for anything suspicious like viruses or bacteria.
Precisely. And what's fascinating is that using immunopeptidomics, they found a significantly higher percentage of NCORFs. It suggests that many of these NCORF-derived proteins are being translated but then rapidly degraded, making them hard to spot with traditional methods, but visible to the HLA system.
So it's like these microproteins are playing a game of hide and seek with scientists. They're fleeting and elusive, but the HLA system catches them in the act.
A perfect analogy. And this discovery has huge implications, particularly when it comes to understanding the role of NCRFs in disease. And their potential for new treatments, especially in the field of immunotherapy.
Okay, stop right there.
Okay.
Immunotherapy.
Yes.
Now you've officially piqued my curiosity. Tell me more.
Well, immunotherapy harnesses the power of our own immune system to fight disease.
Right.
And the fact that so many NCORFs are showing up in immunopeptidomics data makes them incredibly intriguing targets for new immunotherapy treatments.
Oh, wow.
Imagine we could potentially train the immune system to recognize and attack these previously unknown proteins in diseases like cancer.
That's amazing. So these tiny proteins could be game changers in the fight against some of our most challenging diseases.
Absolutely. And this is where the research you sent takes center stage.
Yeah.
They analyzed this massive amount of data and found evidence for thousands of potential new proteins originating from these NCORFs.
That's incredible.
It's a whole new frontier in biology.
Wow.
Yeah. And this research goes beyond just identifying these NCORFs.
Yeah.
They've created a system for organizing all this information.
Oh, wow.
Something called Peptide Atlas.
Peptide Atlas.
Yeah.
That sounds like a treasure map for the world of proteins.
You could say that Peptide Atlas is a public database that catalogs all the evidence for protein existence. It's like a central library where
Yeah.
scientists can go to verify their findings and share new discoveries about proteins, including these NCORFs.
Oh, wow.
This research added a whole new layer of information to Peptide Atlas, making it an even more valuable resource for scientists studying these tiny proteins.
So it's constantly evolving, just like our understanding of NCORFs.
Exactly. But even with all this data at our fingertips, determining whether an NCORF should be officially classified as a protein-coding gene
Right.
is still a complex process.
Didn't they develop a system to help with that, some kind of ranking system for NCORFs?
Right. They came up with a 5-tier system
Okay.
to classify NCORFs based on the strength of the evidence.
Got it.
Think of it like a ranking system, with Tier 1A being the gold standard.
So Tier 1A NCORFs are the ones with the most solid evidence backing them up.
Exactly. Tier 1A means multiple high-quality peptides have been detected in conventional proteomics experiments.
Right.
And there's also supporting data from something called ribosec.
Hold on. Remind me what ribosec is all about.
Okay, so ribosec is a technique that lets scientists see which parts of RNA are actually being translated into proteins. Remember, DNA holds the blueprint, but RNA is the messenger that carries those instructions to the protein-making machinery of the cell.
Right.
Ribosec allows us to see which messages are being read and turned into proteins.
So Tier 1A candidates, they've passed all the tests with flying colors.
Yes.
They've got solid evidence from multiple sources.
Precisely. And a few of these high-confidence Tier 1A candidates have already been added to the GenCode database. That's like the official registry of human genes.
So these microproteins are finally getting the recognition they deserve.
They are.
But what about the other tiers?
Okay.
Are they still important?
Absolutely. Especially when you consider immunopeptidomics.
Right.
Remember, this technique is incredibly sensitive for detecting NCORFs.
Yeah.
Even if an NCORF doesn't quite make it to Tier 1A status,
Okay.
it could still be biologically relevant, especially for immunotherapy.
I'm starting to see how this all connects. So even if we don't have a complete understanding of an NCORF's function, the fact that it's showing up in immunopeptidomics data makes it a potential player in the immune system's game.
That's a great way to put it, and it raises a really interesting question. How do we decide when an NCORF deserves to be called a gene?
Yeah, that's a good question. What are the criteria?
Well,
Just proving a protein exists isn't enough, right?
you're right. It's not that simple.
Okay.
There's debate about whether we need to demonstrate that the protein has a specific function
Right.
before we officially declare it a gene.
Okay.
It's like finding a brand new tool in a toolbox,
Awesome.
but not knowing what it's used for.
Right.
We can see it's there,
Ah.
but we need to figure out its purpose.
Okay, so this is where things get a little tricky.
Yeah.
How do scientists determine the function of these microproteins, especially when they're so new to the scene?
It's a huge challenge.
Yeah.
Remember, these NCORFs are often very small,
Yeah.
and the proteins they produce are often fleeting,
Right.
making them difficult to study. But scientists are developing new tools and techniques to unlock their secrets.
Wow.
And there are already some fascinating case studies emerging.
Oh, do tell. I love a good scientific mystery.
Well, there's one case in particular that caught my eye.
Okay.
It involves an NCORF called C17-NREP146.
Oh,
Quite a mouthful, I know.
I'll take your word for it.
Okay, so what's so special about this C17-NREP146?
Yeah.
Well, this NCORF is what I called a UOORF.
Okay.
That means it overlaps with another gene called PSMC5.
Okay.
And get this, the NCORF actually shows a much stronger ribosec signal than the canonical gene.
Wait, so this little NCORF is stealing the show from the bigger, more established gene.
It really is. And to add to the intrigue,
Yeah.
they found 8 distinct peptides from C17-NREP146 presented by the HLA system. That covers a significant chunk of the NCRF's sequence.
8 peptides.
Yes,
That's pretty compelling evidence that it's actually being translated into a protein.
it is. But here's the catch. Despite all this evidence, C17-NREP146 hasn't been officially annotated as a protein-coding gene yet.
Why not? Seems like it checks all the boxes.
The main reason is that we don't yet understand its function.
Oh.
And that brings us back to a key point.
Okay.
The distinction between protein identification
Uh-huh.
and protein coding gene annotation.
Right.
We can identify a protein,
Yeah.
but figuring out whether it plays a functional role and deserves a spot in the official gene catalog is a whole other challenge.
Right.
It's like finding a mysterious artifact in an archaeological dig.
Right.
We know it exists, but we need to study it carefully to understand its purpose and significance.
I see your point. Yeah. So while we're making incredible progress in identifying these NCORFs and the proteins they produce,
Yes.
there's still so much to uncover about their functions and what roles they play in the grand scheme of things.
You've hit the nail on the head, and this is where the field is heading now.
Okay.
Scientists are racing to unlock the functional secrets of these microproteins. It's like a whole new level of the game has just been unlocked.
This is also fascinating, but I have to ask, what are some of the biggest hurdles scientists face in this NCRRF research?
Okay.
What's standing in the way of these breakthroughs?
Well, one of the biggest challenges is the fact that these microproteins are so small.
Okay.
Traditional methods for verifying protein existence often require the detection of at least 2 peptides from the protein.
Yeah.
But many NCRFs are so tiny that they simply can't produce 2 peptides that meet the current standards.
So we might need to rethink our criteria for protein identification to accommodate these small but mighty microproteins.
Exactly. And then there's the question of whether HLA immunopeptidomics data alone should be considered sufficient evidence for classifying an NCRRF as a protein coding gene.
Right.
After all, the HLA system is designed to detect and present protein fragments.
Right.
So if we're finding NCRF-derived peptides presented by HLA, shouldn't that be a strong indication that they're genuine proteins?
It makes sense to me. But I suppose there's always the possibility that the presence of a peptide doesn't necessarily mean the protein it came from has a specific function.
That's the debate.
Yeah.
And it gets even more complicated when you consider the source of the data. A lot of the current research on NECORFs comes from cancer samples and cell lines. So if a protein is only detected in cancer cells, is it a cancer-specific product, or is it simply more easily detected in that context?
That's interesting.
It's a question scientists are still grappling with.
It seems like the origin of the data can have a significant impact on how we interpret the findings. Yeah. It's like trying to solve a puzzle with pieces from different boxes. It might all fit together.
Yeah.
But is it the right picture?
That's a great way to think about it. And then there's the issue of evolutionary conservation.
Refresh my memory. What does evolutionary conservation mean again?
Essentially, it means that a gene or protein sequence has remained relatively unchanged over millions of years of evolution. This suggests that the gene or protein plays an important role.
Got it.
As natural selection would have weeded out any harmful mutations.
Okay.
If a gene or protein is highly conserved, it's probably doing something crucial.
Like a tried and true recipe passed down through generations. It must be good if it's lasted this long.
Exactly. But here's the thing. Most of these NCRFs are evolutionarily young, meaning they haven't had much time to accumulate changes.
Okay.
So we can't rely on evolutionary conservation to tell us how important they are.
So it's like trying to judge the value of a brand new painting. We can't compare it to the masterpieces of the past because it's just too new.
Precisely. We need to develop new methods for evaluating these young NCRFs and deciphering their roles in the cell.
So it seems like the world of NCRFs is both incredibly exciting and incredibly challenging.
It is.
Scientists are on the brink of groundbreaking discoveries.
Yeah.
But they're also facing some major hurdles.
Yes.
What's next for this field? What are the big questions that researchers are tackling now?
Well, for one, they're trying to develop new tools and techniques specifically designed to study these tiny, often unstable proteins.
Right.
And they're exploring the potential of NCORFs as targets for new immunotherapy treatments. That's a huge area of interest right now.
Right.
But ultimately, the biggest challenge is cracking the code of their function. We're starting to get hints that they might play roles in a wide range of cellular processes.
Yeah.
But there's still so much we don't know.
It sounds like we've only just begun to scratch the surface of this hidden universe within our DNA.
Exactly. And the more we learn about NCORFs, the more we realize how much more there is to discover.
Right.
It's a truly exciting time to be in this field.
And you know what blows my mind about this whole thing? This research you sent, it suggests there are even patterns to which NCRFs are easier to find.
That's right. They actually found some intriguing clues about what makes certain NCRFs stand out from the crowd. For example, the NCRFs that were detected, they tended to be longer and have a higher isoelectric point than the ones that went undetected.
Isoelectric point. Remind me what that is again.
Right. It's basically a measure of a protein's charge.
Okay.
And it seems that a higher isoelectric point might make an NTRF-derived protein more likely to be presented by the HLA system.
So it's kind of like having a VIP pass to the cellular security checkpoint.
Exactly.
Making them easier to spot.
Right.
So not only are these NTRFs tiny and hard to find, but some are even sneakier than others.
Exactly. But the researchers uncovered another interesting pattern.
Okay.
The genes that give rise to these more detectable NTRFs, they tend to be more highly expressed.
Okay.
Meaning they're actively producing more RNA transcripts.
That makes sense. If a gene is churning out more RNA transcripts, there's a higher chance that its NTRFs will be translated into proteins, making them more likely to be detected.
Precisely. And this highlights how crucial it is to combine information from different sources.
Okay.
Proteomics, immunopeptidomics, ribosec, and transcriptomics to understand the full story of NTRFs. It's like piecing together a complex puzzle with each piece of data providing a unique clue.
I love that analogy.
Yeah.
It really paints a picture of how much detective work goes into this research.
It does.
So where do we go from here? What are the big mysteries that scientists are racing to solve in this world of NCORS?
Well, one of the most pressing questions is figuring out the function of all these newfound microproteins.
Right.
We're starting to see hints that they might be involved in all sorts of cellular processes, from regulating gene expression to interacting with other proteins.
Okay.
But there's still so much we don't know.
It's like having a toolbox full of shiny new tools, but no instruction manual.
Exactly. Another major area of focus is exploring the potential of NCORFs for immunotherapy.
Right.
Remember, the fact that so many of them are presented by the HLA system makes them incredibly promising targets for new treatments.
It's exciting to think we could be on the verge of a whole new generation of immunotherapy treatments, all thanks to these tiny proteins.
Absolutely. Researchers are already making strides in this area, but the biggest challenge remains deciphering the functions of these microproteins and how they impact our biology.
Wow. It's amazing to think that all this time there's been this hidden world of tiny proteins operating within our cells.
Right.
And it's only now that we're beginning to uncover their secrets.
Yeah.
So what's the final takeaway for our listener today?
This research is a powerful reminder that we still have so much to learn about the complexity of the human genome. These NTRFs were hiding in plain sight, and their discovery has opened up a whole new frontier in biology. Who knows what other mysteries are still waiting to be unlocked?
It really makes you wonder what other hidden treasures are lurking within our DNA, just waiting to be discovered. I think this deep dive has left us with more questions than answers, but that's the beauty of science, isn't it? It's a never-ending journey of exploration and discovery. And I, for one, can't wait to see what the future holds for this world of NTORFs. Thanks for joining us on this deep dive.